We're really excited about today's event as it's our first digital event sharing the impact of our research programme. Last month we published our first impact report to really understand and assess how the funding we provide for ovarian cancer research is making a difference, and we're delighted that so many of you could join us today to hear from our speakers. Before we kick off, I'm Helen Dickens. I'm the Director of Programmes at Target Ovarian Cancer, and I'll be chairing today's session. We're really delighted to be joined by Professor Richard Edmondson today. And while unfortunately due to other commitments, Professor Ruth Plummer is unable to join us live, she's recorded a fantastic presentation to share with you this afternoon. We do have quite a packed agenda and we will run through swiftly to make up for the time that we've lost at the start. So I'll give you a very short overview of our research programme and then we'll hear from Professor Plummer with her perspective as the chair of our Scientific Advisory Board. Professor Edmondson will then provide a deep dive update on the work his team have been delivering at the University of Manchester, and then he'll stay with us for a short Q&A at the end. And I know lots of you will be looking forward to hearing his answers about some of the hopes and futures for ovarian cancer research. Just want to run through a bit of housekeeping. You may have noticed that Zoom looks a little bit different if you've not used Zoom's webinar feature before. We're using this today because we have got a large number of you attending, which is fantastic. And we wanted to make sure that everybody was able to view it as easily as possible. Because we are using the webinar feature, that means that while you can see us, your camera and audio is not being streamed to other attendees today. However, the chat box is available. So if you do need anything, if you have any questions as we go through, any technical issues or any questions on the presentations, a member of the team will be monitoring this throughout the session. Be aware these are only viewable by our speakers and the Target Ovarian Cancer team, so please do share what you need with us. As Kate mentioned, we were hoping to stream the event live through our ovarian cancer community on Facebook, and we will look to make sure that this is shared there as well. So anybody watching back on the session, a huge welcome. We will be recording the event. We hope you'll be able to stay with us for the full session, but if you need to step away, we will be sharing that recording if there's anything that you missed today. When we come to the Q&A, I just want to say a big thank you for everybody's questions that you've submitted in advance. We've got a huge array of questions, really important questions as well, and we've shared those with Professor Edmondson for the Q&A, and we hope to cover as many as we can. Please do be aware though that we may not be able to comment on some of the questions which are very specific to an individual's treatment or personal circumstances, and there may be some that we won't be able to cover today. But if you do have any further questions, or if anything is discussed today that causes you any concern, please do call our support line staffed by our fantastic nurses. The support line is open every day during the week, 9 AM to 5:30 PM, and that number is 020 7923 5475. And my colleague Kate will pop those details into the chat so you have them to hand if you need them. Well, I just wanted to kick off our event today with a short overview of Target Ovarian Cancer's role in ovarian cancer research. Many of you will be aware of this already, I am sure, but it's just great for us to step back and reflect on what we've achieved so far. So at Target Ovarian Cancer, we've been funding research for the last 12 years through our Transformational Research Awards programme. It's a bit of a mouthful, but what does this mean? It's really an approach that involves scientists and clinicians working together closely so they can share insight and ideas on what causes ovarian cancer and how we can best treat it. And through this programme, we've awarded over £1.5 million so far to 10 groundbreaking projects across the UK. And we really focus on research that we know will benefit the largest proportion of women affected by ovarian cancer in the shortest time frame, with a particular focus on improving treatments. But it's not just about the projects that we fund. It's really important to us that we are able to support the research community to drive even further research into ovarian cancer, and really build capacity into that community by supporting innovative projects that open new routes for research, and really importantly through supporting researchers who may be early on in their careers so that they're able to play a role in ovarian cancer research for many years to come. We also support our researchers to share their work with the wider community through events like today, and hopefully go on to achieve more funding as well. So we won't have time today to go into lots of detail of all of the research projects we funded over the last 12 years, but really excited to have Professor Edmondson here with us today to talk in detail about the project that he and his team have been working on. And there's plenty of other highlights if you look on our website of other projects that are focused on genetic testing, bowel obstruction and diet, clear cell ovarian cancer, low-grade serous ovarian cancer, and also effective treatments. So please do make sure that you're having a look at those. We know that these projects are driving real change in ovarian cancer research. We take a gold standard approach to assessing the projects that we fund. And as a member of the Association of Medical Research Charities, the AMRC, we apply best practice in securing and ensuring that only the highest quality grants are supported by our funding. We're joined in reviewing those applications by our incredible group of research advocates, and I know many of you are on the call today. These are our volunteers who support ovarian cancer research in so many different ways, and it's really important to us that the needs, views and hopes of everyone affected by ovarian cancer are really central to the research that we fund. And together our scientific advisory board and our research advocates apply a really rigorous peer review process with lots of scrutiny to anybody who applies for one of our grants, making sure that we only fund the best projects. So I'm now delighted to introduce Professor Ruth Plummer, who's the chair of our scientific advisory board, to tell us a little bit more about all of this. Professor Plummer is Professor of Experimental Cancer Medicine at the Northern Institute for Cancer Research at Newcastle University and has chaired our scientific advisory board since 2017. Unfortunately, as I mentioned earlier, due to unavoidable commitments, Professor Plummer is unable to join us live, but she's kindly recorded a short presentation to tell us a bit more about the role of the Scientific Advisory Board and the impact of the research programme to date.
Well, thank you for joining today's event, and can I start with an apology that I'm actually not able, due to scheduling and clinic, to actually join you live, but I was very keen to join the event virtually and explain to you a little bit about the Scientific Advisory Board, which I chair for Target Ovarian Cancer. So I'm Professor Ruth Plummer. I'm a medical oncologist and work as run an early phase trials unit, but I'm chair of the Scientific Advisory Board. What I was going to explain to you is how we look at projects, assess them for research funding, try and outline the impact of the research to date, although I know you're getting a lot more detail on that and have the document, but also talk a little bit about the importance of the research programme that Target Ovarian Cancer is able to run. I mean, important challenges for research in ovarian cancer are finding new drugs and getting new targets and targeted treatments. Also understanding why tumours develop resistant to therapy. A lot of our patients respond to carboplatin, but then the tumour becomes resistant. Also looking at new immunotherapy techniques and approaches which have worked really well in other cancers, but not as well in some of the major cancers, including ovarian cancer. And that understanding why treatments work, the effectiveness, and then how do we build on that? And also prognostic markers so we can see who to treat, that mantra of the right drug for the right patient or the right tumour. That's what we would like to be able to do in cancer research. Whenever we do any research, people have bright ideas, but the gold standard is peer review to try and, with the limited funding that's available, and we know the funding space at the moment is really challenging, how do you choose the best projects to go forward? And that's done by peer review. And how it's done at Target Ovarian Cancer is that researchers across the UK are invited to submit proposals in line with the stated research strategy. And then we review those as a scientific advisory board, thinking about, is this addressing a critical either scientific question or a clinical gap? Is there the potential for it to be innovative? How likely is it to achieve what is being set out in the programme? And are the investigators proposing it, have they got the skill set? Have they got the right collaborations and links? And also, because resource and money is precious, are they actually justifying carefully what they want to spend and how much it'll be? So I'm not alone. We have a scientific advisory board, and out of it, actually, I am the one who at the moment does the least work in ovarian cancer. I run an early phase trials unit running trials of experimental drugs, and that was felt important by Target Ovarian Cancer in that I'm giving an oversight, but I'm not conflicted because I'm not so close in the area. But obviously we need experts in the area, and we've got Professor Richard Edmondson, who I know is talking later, but he's a clinical professor, but he's actually a gynecological surgeon. There's Ros Glasgow, who's a consultant medical oncologist like myself, but works up at the University of Glasgow, running a program into ovarian cancer research. There's Maurizio Dincalci, who's clinically qualified, but now works in cancer pharmacology, working out new drugs. And again, his area of interest is gynecological cancers. Rebecca Crystallite at Guy St Thomas's, another consultant medical oncologist who treats ovarian cancer. And Chris Lord is a very smart scientist who works at the Institute of Cancer Research in London and joined the board to give us that scientific breadth when we're assessing proposals. And research impact, which is what the meeting is about today, it is a bit of a long road. If we think of it in years, what is the impact? And for me, it's what we do for patients. And when I'm asked about, am I proud of what's being done on the trials unit? And we've been involved in developing some new drugs, including rucaparib, one of the PARP inhibitors used in ovarian cancer. I dosed the first patient and wrote the prescription for the first patient in the world ever to get a PARP inhibitor, back when this drug was at an earlier stage. But actually, for me, the impact of what I do is not those big projects, it's those individual patients where, they've talked about milestones in their lives that they've wanted to see, and we've been able to help that happen. But that impact takes a while. With a project starting, you've then got the research, which is 1 to 3 years. You then need to write up the report, check the data and publish it, which can take up to another year. And then realistically, to get a clinical impact in patients, you need follow-on funding. So we are adding to the years, and almost the frustration when you've got a good idea of how do I get this to patients and how do I get the benefit there. But the Target Ovarian Cancer funding has been fantastic in terms of the impact. If you think of a traditional scientific impact in terms of those publications, those outputs, there've been, some really high profile in Nature Communications, top level journals of the work that's been funded by Target Ovarian Cancer. One of the other things we look at is when we're trying to assess impact is that follow-on funding. And for at least every pound that's been spent on research, researchers have secured the same amount or more in further funding from other sources to keep the research going. So you've got that added value. So that's the way of looking at the impact to date. And if you think about the importance of the research program, for me, it's also the breadth that it spans, because when I was looking through the impact document recently, there's the science, there's the exciting work that was done with circulating tumor DNA, little bits of broken DNA that all of us have got in our blood. But if patients have got a tumor on board, you can find little bits that must have come from a tumor. And we've had researchers trying to look at how specifically they can find that, and can they find something that might help for early detection of ovarian cancer. We've also, going right to the other end of the patient journey almost, we funded some research where they've set up a genetic counselling model to try and smooth that pathway and get a sort of seamless, the BRCA testing and how you counsel patients about a familial risk of cancer. So a very different thing. But then obviously dear to my heart, it's the novel treatment and the work that we funded where combining chemotherapy to try and make immunotherapy more effective the researchers who did that are now trying to build on that into a new trial. And then one of the other things that I feel is really important is looking at the partnership when people are proposing grants, but we also within the strategy really encourage that if the grant is not coming from an early career researcher, that they should be included. We need that next generation of researchers. So it's how do we do that? And the other thing that Target Ovarian Cancer has done strategically is, for example, partnership with the MRC, and they'll help with some of the peer review for one of the PhD fellowships that's going to be funded. So there's that element of the partnership as well. So overall, I hope that gives you a flavour of what the Scientific Advisory Board tries to do to support Target Ovarian Cancer's mission to find, better treatments and better ways of looking after patients with ovarian cancer.
Thank you to Ruth for sharing that perspective with us this afternoon. And I think it really showcases some of the impact that our research programme has had to date, as we look at the number of publications, the impact that we've had on future research, and particularly seeing the researchers that we fund going on to continue their careers within ovarian cancer research and new projects being funded is absolutely fantastic to see. As I said previously, you can find out more information about the research that we funded, and if you haven't seen it already, please do download the impact report on our website. We'll pop a link in the chat so that you can follow that up after the event. So we've talked in broad terms about the impacts of our research, but I think what we all want to hear is to really understand a little bit more of the detail. So I'm really delighted now to invite Professor Richard Edmondson to present to us. Professor Edmondson was appointed as a consultant in Gateshead, an honorary senior lecturer in Newcastle in 2005, and in January 2014 he took up the chair of gynecological oncology at the University of Manchester. He'll be providing an update on the exciting project he and his team have been leading, looking at developing more personalised approaches to treating ovarian cancer. So Richard, over to you.
Fantastic. Thank you so much, Helen, for the invite. Let's see if I can get these slides sharing. Can you see my first slide now? That should be working for everybody. And thank you for inviting me to speak today. And I just had a quick look down the participant list and I recognise a few names there, so it's really good to see some familiar faces. But for those that I don't know, it's nice to meet you all. I'm Richard Edmondson. As everybody said, I'm a professor in Manchester, and I used to work with Ruth up in Newcastle sort of back in the day. What I thought I'd do in the next 15 to 20 minutes is try and take you through a little bit of the work that we've been doing in Manchester based using the Target Ovarian funding that we received 3 years ago, but also try and use it as an opportunity to try and show you a little bit about the research process and some of the progress that different groups have been making in different areas, and the way that we approach some of these problems to see if we can ultimately get better treatments and improve care for patients with ovarian cancer. So I'm doing this talk on behalf of my lab group, but the one person I do want to mention is Tom Walker. Tom's on the call today. Tom has done a lot of the work that I'm going to describe today, and he's going to be slightly horrified that I'm going to try and précis 3 years work into about 3 slides. I can't stress enough the amount of work that he's put into this project in getting the results that I'm going to show you, but it's nice to have him on the call today as well. So Ruth has already hinted and sort of outlined the fact that Target Ovarian Cancer has now been sponsoring research for several years, the best part of a decade, and overall they've funded 10 different projects, and this map just shows you where those projects are. And it was just very interesting looking at them and sort of pulling them apart. I began to realise that they really do cover the spectrum of ovarian cancer. So 2 of those projects, Lizzie Moore's project and Mark Tishkovitz, both in Cambridge, have really focused on prevention and early detection, whereas probably my project and Martin Miller's have both looked at the management of the early management of disease. And then there are 2 further projects looking at the late management of disease, and particularly looking at resistance to chemotherapy. And then Anne Lansley's and Aggie Michael's projects have both looked very much at symptom control. And then the sort of Scottish contingent of Ian McNeish and Charlie Gourley have both looked at different types of ovarian cancer. So you can see straight away there's a real mix and wide breadth to the programme that's been funded by Target Ovarian Cancer here. And I think that is a real testament to the charity in that it's looked at lots of different ways of funding different parts of the jigsaw that is the disease and how we manage it in the future. So I'm going to focus a little bit on our project, really, because that's obviously the one I know most about, but just that is a reminder that there's an awful lot of other things going on or have gone on as a result of the funding that's been put out in the past. So just by way of background, to try and introduce you to the project that we've undertaken and what we've been doing, I just thought I'd show you this. So welcome to a little bit of the human genome. What I'm showing you here is a little bit of the code that is written in your DNA that forms your instruction book that is in every single cell in your body. Now, if you look closely, you'll see that that's just a series of letters, and if you pay attention, you'll see that there are really only 4 letters there, T, C, G and A, and that is the language of your genome. It's all written in 4 simple letters, T, C, G and A. Now, when you look at it there, it's not the most exciting read, I have to say, because it is just strings of characters. And you'll see there are quite a lot there, but your genome is actually very, very big. So that's just a little bit of it. If we zoom out a little bit, you'll see that it doesn't just cover a single page. In fact, it doesn't cover 2 pages. And bear in mind, that's all written at size 4 font. It's tiny writing. In fact, it doesn't even fill 1 book. That is your whole genome written at size 4 font of just T, C's, G's and A's. And that's the instruction book that is contained in every single one of your 17 trillion cells that tells those cells how to form and how to make themselves. That's amazing enough. But the other bit of the story is that that genome is being looked after in every cell by, let's call them librarians, a series of librarians who are running around the genome, looking for any mistakes, any errors, and putting them back in the place they should be. And just like librarians, they're looking for books that are out of place and putting them back into the right place. They're looking for books that have been put upside down or in the, you know, slightly out of line, and they're lining them all up and straightening them up and making sure that your genome is pristine in every single cell and doesn't vary. And that system works very well, but sometimes it goes wrong. And when it goes wrong, there's the ability for mistakes to creep in, and gradually the genome becomes littered with more and more mistakes, to such a degree that the cell no longer knows how to behave itself and starts to misbehave and grows out of control and becomes a cancer cell. And cancer cells really are just cells that have grown out of control because they have so many mistakes in their genome that they're no longer behaving properly. And to give you some examples of what we're talking about, I'm going to show you this plot here, which is rather a technical plot, but I think is really interesting. These are called circus plots, and in circus plots, the genome, which is broken down into 23 different volumes, is arranged around the outside of a circle. And every time there's a mistake in that genome, it's just annotated with a series of little letters. So every time you see a little bit of writing around the outside of that genome, it indicates there's a mistake in the genome, a mutation or some other problem. Now, the 2 circus plots you're looking at the moment come from a patient who had, or 2 different patients who have leukemias, and you can see there are a lot of mistakes there in each of those different cells. But now I'm going to show you the same plot, a circus plot for a high-grade serous ovarian cancer, and just look at the comparison. In comparison to those leukemic cells, there are literally hundreds and thousands of different mistakes going on in that genome. And you can begin to see the problem we're up against of working out what's important in driving that cancer cell, what are sort of what we call passenger mutations that just float there, and how we make some sense of it. But the real message I want to get across here is there are lots of different mistakes, but look how different those 2 different cancers are. And we begin to realize straight away that cancer is not just one disease, it's a whole series of different diseases, all have the same underlying problems in their DNA, but those different mistakes can cause the cancers to behave in different ways. It also got us thinking a lot about how do those mistakes accrue, because in normal cells, those librarians that we talked about are running around repairing the DNA. So almost you can guess that those librarians must be broken or missing in some of these cancer cells to allow those different types of mistakes to accrue. So let's just think about those librarians in a little bit more detail. And in fact, when we drill down and work out what's happening in a cell, we know there are 5 different types of librarian, and they're listed here on the right hand side by these rather awkward abbreviations: MMR, NER, BER, HR and NHENG. Sorry, I didn't invent the names, but those are our 5 librarians, and they run round repairing the genome, but they all have a different job and they do different things. So going back to our library analogy, one is responsible for putting books back onto the right shelf, another is responsible for turning them the right way around, another one is making sure they're sort of not back to front, and all those sort of different things. So different mistakes in the genome are repaired by these different 5 processes or librarians. And as we got to think about how a cancer cell could generate itself and actually survive, we guessed that at least one of those librarians, or those mismatch repair, or sorry, DNA repair pathways, must be broken in every single cell. So we came up with a hypothesis, and our hypothesis was that all cancers will have a defect in their ability to repair DNA. So they will all have at least one broken librarian. And secondly, we hypothesized that the pattern of those defects would determine the behavior of the cancer, and that might be useful in determining how we treat the cancer. So we wanted to do 2 things to with this hypothesis to understand what the mistakes were in the cancers, and secondly, define whether that could predict the behavior of that particular cancer. So that was our hypothesis and our theory. And that's the bit of research where you sit and you spend a bit of time thinking about this and chewing it over and coming up with ideas. And it's a really fun part of the research process. But research is more than just coming up with an idea. And in many ways, the principles of research can be highlighted on this very simple schematic of a cycle, that once you've generated a hypothesis, you then need to set about trying to test that hypothesis and see whether it's true or not. It's fine having an idea, but you've got to try and establish whether that idea actually is true or not. But to test a hypothesis, you have to take an interim step and develop a model. And in scientific research, we all develop a model that we can use to test our hypothesis. And I'll show you some ideas of models in just a second. Once you've tested your hypothesis, you then have to scrutinize your data. Now, Ruth mentioned in her presentation the idea of peer review for approving grants, but also peer review is the process where we publish papers and put them out there and expose them to the public and to the research community, who can then criticize them, comment on them, pass judgment on them, and then we use that to then go around the cycle again and generate further hypotheses and develop the next research idea. I didn't put on this cycle, but there's a very important part of this cycle, which is also the spin out, that we hope that by the time we've scrutinized data, new treatments and new ideas for therapies are coming through and being able to develop into practical changes. But nevertheless, that's the research cycle. So if we've generated our hypothesis, the next thing we really need to do is try and develop a model. Now, there are lots of different models that are used in cancer research. And traditionally, most of those were carried out by what we might call discovery scientists. The traditional, if you like, boffin sitting in his white coat in the lab, well away from any sort of patient and clinical interaction. And discovery scientists are absolutely fundamental to the process of research, because without the stuff that they find out, we can't move that through into clinical improvements. But discovery scientists have tended to use models such as mice or other animals, or cell lines, which are cells grown in the laboratory over long periods of time. And the reason they use these models is they're very reliable and they're very stable. They don't change with time, and so you can use them over a long period of time and do lots of different things to these. They're very manipulable, and that allows you to test different things in different ways in the laboratory, and extract DNA and test that in different ways. So discovery scientists have tended to use those sort of models well away from patients. On the other end of the spectrum, clinical research is the sort of research that Ruth does in her early phase trials unit, which is largely using patients as the model. So testing things in a patient environment. And that's really, really useful. And that is a gold standard of how things get into clinical practice. But it's very difficult to do, because without doubt, you've got to know that the thing that you're trying to give to the patient is very safe and is very likely to work before you get to the point of clinical implementation. So it's very much an end part of the research process. And for many years, we had this slight disconnect with what the discovery scientists were discovering in their labs and what we were able to implement into clinical trials and therefore new clinical treatments. And that space needed to be filled by something, and it was filled by what we call translational research. And if you look on the Target Ovarian Cancer website, you'll see that they actually say that what they fund is translational science. And all of those 10 projects that I highlighted to you earlier can arguably fit into this space of translational science, where you take what the discovery scientists have told us and we try and move that through into the clinical arena that can then go into clinical trials. And there are hundreds of different ways of doing translational research and different models, but I've outlined here the model that we use because we find this particularly useful. So we do still involve patients, but in our research model, once we've identified a patient, we actually take a part of their tumour, and we take that at the time of surgery, and we take the tumour into our laboratory, we break it up into small pieces, and we start to grow it, and I'll show you what I mean by this, in the laboratory environment. So we have growing cancer cells in our labs that we can then use for our studies. And really importantly, we can link that back to the clinical data about the patient. So we can understand what the cancers are telling us in the Petri dish, but also link that to how the patients are doing and how they've responded to therapy and what the effect of various therapies have been. So this is a really useful model that we can study cancer and link it back to patients, even though we're not actually doing anything different to the patient themselves. It is a rather complex process to do. And it relies on a few things. You've got to have a lot of regulatory processes in place, but that's just some boring paperwork that we'll get through. But the really key thing that you need to have is you need to have your operating theatres, or your source of tissue, if you like, which in our case is operating theatres, very close to your research labs. And we're really lucky in Manchester because we've got, my research labs are just 3 floors up from our operating theatre. So I tend to operate on a Thursday, and when I'm operating, tissue can be removed from a patient as part of their routine operation, and it can go straight into the research laboratory and be used in the lab studies at that point, very quickly and very efficiently, and can be moved around the hospital without impacting on the patient at all and without impacting on their care at all. But they're able to provide us with tissue, which is incredibly useful for what we're doing. So let me show you what I mean by that. So here's some pictures of what we mean by our translational model. So I want you to just look at the 3 boxes at the top, the 3 sort of grey squares. And what you're looking at there are ovarian cancer cells that have been taken from a patient who was having surgery and brought into the laboratory. They've been broken up into a single cell suspension. So the cells are floating around one by one, and then they're put into Petri dishes and grown in a Petri dish. On the top left, you can see what the cells look like just 24 hours after they came out of the patient, and they are alive and well on that plastic. And we have a slightly perverse relationship with these cells. As a patient or as a clinician, our focus is really on trying to make sure that cancer cells die. All we want to do is kill cancer cells. But as soon as I get into the laboratory, I change my mind, and all I want to do is try and keep these cells alive. So my laboratory team spend their life getting very excited when they can keep cancer cells alive. But as a clinician, I really want to have the opposite effect and kill them. But you can understand that dilemma. But they're alive and well at 24 hours. In the middle one, that's 72 hours, and they're still nicely growing. You can see they're covering the bottom of the Petri dish. And on the right-hand side, you can see at 2 weeks, we've managed to split them. What we've done is we've lifted them off the plastic, we've divided them into 2 or even 4, and then put them back into 4 Petri dishes and growing them that way. So they will grow for 2 or 3 weeks before they naturally go through a process that we call senescence, where they actually die. But we have them alive and well for about 2 to 3 weeks and can study them at that time. It's really important that we know what we're growing, and are these really cancer cells? And you could turn around to me and say, I don't know what those are. Those could be any old cells. But we do a whole series of tests, which is what the pictures are at the bottom, showing you different things we look for in those cells to make sure that they are the cancer cells that we expect. And I'll just draw your attention to the bottom right one, the CA125 panel. Most of you will know about CA125. It's a marker that we use in the blood, but the cancer cells actually express it. And if they express it, they go red under this particular test. And you can see those cells are all bright red, which means they've got plenty of CA125 there. So they are good, in inverted commas, healthy cancer cells doing what they're supposed to in our lab. I'm just going to take a slight digression now just before I tell you about the rest of the project, just to show you some data, because I thought you might be interested in this. I'm very excited about this, and we only picked this up in the last week or so. So these are brand new pictures, and virtually nobody else in the world has seen these. We're very interested, obviously, in the idea that ovarian cancer probably doesn't come from the ovary, and we think a lot of ovarian cancers probably start life in the fallopian tube. So to try and study that a little bit further, we thought we might be able to try and grow normal cells in our lab as well as the cancer cells. So here, what you're looking at is a microscope image of a tiny little piece of fallopian tube that we've taken at the time of surgery, and we've put into a dish of chemicals which will break it up into that single cell suspension. On the same day, we then plate that out, and you can see there are tiny clumps of cells here. By day 3, day 4 and day 7, you can see they're growing into these lumps of cells, and by day 11 they're growing into these really nice little clusters of cells. Really excitingly though, they're actually growing as hollow little spheres, and they're producing fluid into the middle of the spheres, which is exactly the same fluid that you'd get from a fallopian tube. So we're now growing in the laboratory three-dimensional structures that really do represent the fallopian tube, and these are going to be a great model for the future for actually studying how these cancers start, because we can manipulate these in the lab now to try and get some idea of the first steps of cancer. So that's a digression, but it just shows you that with this idea of taking tissue from patients at the time of their surgery, we can use that to do all sorts of things in the lab. So back to cancer, because that's really what this story is about, and about our patient samples. It's fine now, we've got a model. Remember, we've got a hypothesis. We want to look at what these librarians, these DNA damage repair pathways, are doing in the cancer cells, and we've got a model to study it. So how do we set about doing the next bit, which is to test that hypothesis? So we built a really quite a simple idea that if we could take some patient samples, could we get those growing in the lab, and then could we run them through a series of tests? And these tests would look at each of those DNA damage repair pathways and try and assess whether they were working or not. And these are very much functional assays where we want to stress the cell and induce some of the DNA damage that they would normally be exposed to, and then see what happens. So going back to our analogy, we're going to go into the library, we're going to disrupt it, we're going to take some books, throw them all over the place, and then see how the cell repairs them and which methods it's got to repair them. Can it get the books back on the shelf or not? Is it sorting them out once they're on the shelf? Is it replacing bits that are torn and damaged, and so on and so on? So we built, and I'm not going to go through the techniques of how we did it, but we built 5 different assays, one for each of the 5 different repair pathways. At the same time, remember, these samples come from real patients, so we've been able to follow those patients up and see what happened to them, and then try and link those 2 bits of information together and see whether the information we got from our assays links to the clinical outcomes of the patients. So this is a rather technical slide that just shows you some of the results we get from those 5 different assays. So the different pictures show you the kinds of things we're looking at in the lab and the results that come out of the various assays that we've done. But all of them can simply be sort of described as inducing some DNA damage, disrupting that library, and then seeing what happens to the cell after that and how it repairs it. All 5 of those assays have been described by different people, including us, over many years, but nobody had ever really put them all together and managed to do them in the same cancer cell at the same time. And the really sort of clever bit of what we managed to do was manage to do them all on the same patients, or the same patient samples. These assays have generated an absolutely enormous amount of data, and we continue to wade our way through some of that data and make sense of it. I'm not going to show you all of it because it amounts to pages and pages of data, a lot of which is really, really interesting, but I'm just going to show you 2 things really, which are kind of headline summaries from all of that data. And the first is this, which looks like a pretty box, but let me explain what this pretty sort of grid is showing you, and then show you how we're going to interpret it. So each of the vertical bars in this little table, or this little grid, is an individual patient. So we've got each of this is just a subset of our 50 patients, a subset here of all identical patients. These patients all had high-grade serous ovarian cancer, and it was all advanced disease at the time of presentation. So they're all very, very similar patients, but each patient is represented by a vertical line. The horizontal lines represent the results of those 5 different assays. So you can see them labeled down the right-hand side. Essentially, if the box is shaded yellow, that particular pathway is working in those cells. If it's purple, it's broken. If it's green or blue, it's a variation somewhere between working or broken. And I think what I want to just get you to do is have a look at that and not count things, just look at it in general, and you'll see, I think, straight away that first of all, every single cancer has at least one of their pathways broken. There isn't a single cancer there that is all yellow. The one on the right hand side is the closest to being yellow, but that's still got defects in 2 of its pathways. The one on the right hand side has got almost all its pathways broken, but no cancer has all of them broken either. So each cancer has some breaks in its pathways, but not all of them are broken. The second thing I want you to notice is that all of the cancers are different. So remember, I said these are all essentially the same looking patients. They've all got stage 3 or 4 high-grade serous ovarian cancer, but when we drill down into the detail, they're all actually different. They've all got very different mechanisms for repairing their DNA. So as we drill down more and more into cancers, we see that they're actually quite different underneath it. And the last thing I want to just sort of highlight is that the two pathways that are the most different are the HR and the NHENG pathways, and those are both repairing double strand breaks. So they're doing sort of the same thing in different ways, but they're almost mutually exclusive, which we thought was quite interesting. So we end up with this picture that what's supposed to look like very similar cancers are actually really, really different underneath it. And so the next question then we wanted to ask was, is that related to their clinical outcome at all? So, of course, as I said, we've been following these patients up since they donated their samples to us, and we've been able to track them now for about 15 months. And as time goes on, we'll be able to track them further. And what we've been able to show, and these sort of plots kind of highlight that idea, that the different patterns of their DNA damage repair pathways clearly has a relationship to whether those cancers come back, or indeed whether the patient survives or not. And we can now build models or signatures that will predict whether patients are likely to respond to their chemotherapy in terms of how long it is before their cancer comes back, or whether indeed they survive. Now, the numbers are still relatively small here, and there's a lot of validation work to show whether this is a true phenomenon or not. But we think this is really exciting, because this is beginning to say that by testing the tumours in this particular way, we're likely to get a very accurate way of determining whether patients are going to respond to chemotherapy or not. And of course, that's really, really important, particularly in the relapse setting when cancers have come back. When trying to decide whether having chemotherapy or not is a really important decision to make. So it may well be that the assays that we've developed here could be turned into something that would be very useful in terms of predicting whether patients are going to get benefit from their chemotherapy or not. So by asking the question of the basic biology, trying to understand what is going on in the tumours, we've been able to turn that round into something which is very likely to have some clinical impact. So to kind of summarise, I want to just highlight that it's really important that we can use patient samples to study ovarian cancer. And we strongly believe that sitting in this translational space in the middle between discovery science and clinical research, both of which are phenomenally important, but by using patient samples, we can help bridge that divide between those two areas. And then just hopefully I've convinced you that by really trying to drill into each cancer and understand it in a bit more detail, we can find real differences within cancers that are supposed to look actually the same, but those differences are probably really important in predicting outcome. The assays that we've started to put together here might be used to determine clinical management. There's an awful lot of challenge in doing that last bit, because don't forget, we're using cells that are taken straight out of a patient at the time of surgery and brought to my lab, usually within about half an hour of being removed from the patient. That's a real challenge if we want to try and scale that up to something that would be usable in a clinical environment. But nevertheless, you know, there's some early promising steps there that we could do it. So I'm going to sort of finish there really, just by thanking an awful lot of people, because there's been a huge number of people involved in this project over the last three and a half years. Tom has led particularly the second half of this project and has done a phenomenal amount of work in there. Prior to Tom working on the project, we had Zara, Dr. Ferrari, who worked on this for a couple of years before she moved away, and really set a lot of this up. And then there's the rest of my lab team and other people who've been involved in this in all sorts of ways of helping with this project. I've just acknowledged Maddie and Peter, who've been involved in those fallopian tube cultures that I just showed you. But the real stars of this piece are our patients who've donated tumour samples. Now, I have to say, when I speak to patients about whether they would mind giving us some tissue, they nearly all say, you can have it, I don't want it, and just take it out and use it in whatever way you can. But I'm still really grateful for the fact that they are very happy to participate in this research, because we think it's really important. And then lastly, of course, I must thank Target Ovarian Cancer for funding this project. And by Target Ovarian Cancer, I really mean everybody on this call, because by supporting the charity and fundraising for it, that's what allows us to be able to do this sort of work. So hopefully that's given you a taste of what I've done. There is masses of more that we could talk about that's underneath the surface of this, but I think as a top line, that probably tells you the sort of thing we've been doing, and hopefully is useful. But obviously really happy to take questions through the chat, and Helen will organise that in a second. But thank you very much for listening. Thank you.
Thank you so much for that, Richard. A very short run through what I know is a very complex project, but just fascinating to get that insight into the way that the research has come together, and also starting to see some of the benefits that we can map into now people's clinical care, which is fantastic. So thank you. We do have a short amount of time now for our Q&A. If you did have any particular questions related to what Richard's just talked through, please do pop it into the chat. Although we do have lots of pre-submitted questions that we'd like to try and run through, and I'm very aware of time. So this is going to be a bit of a quick fire Q&A, Richard, if that's okay for you. I think also just to say, I mentioned it before, we won't be able to pose all of the questions today, you know, because if it's too specific on a particular area, obviously we need to look at what Richard's specialisms are. But let's kick off and see where we get to. So I think firstly, Richard, a lot of the questions that we received when we think about ovarian cancer research and potential for the future are around diagnosis, in particular supporting people to receive an earlier diagnosis. And I think many of us saw the coverage of the outcome of the recent trial into screening opportunities. Could you just tell us a little bit about what you're aware of on the horizon for diagnosis, and any updates on potential for screening in the future?
Yeah, so many of you will be aware of what's called the UKC TOX trial, which was the largest randomised controlled trial ever run in the UK, actually. 200,000 women went into that trial to look and see whether screening would work or not. And it was a randomised trial, so half of them got screening and half of them didn't get screening. They were followed up for a long time. Unfortunately, there was no benefit seen to those who received the screening, and it was a really well run trial. It was a very important study, and I think, you know, there's a lot to learn from that. It means at the moment we don't have a screening test that we could say works for ovarian cancer, and I think again it highlights the importance of doing research in this sort of area and doing these trials properly. It was very tempting 25 years ago to think, let's just do scanning and CA125, it'll work, but it was the trial that told us it wouldn't work, and it meant we had to go back to the drawing board. I think part of that is because we need to understand where the cancer is coming from, and therefore how we can actually screen it at a very, very early stage, hopefully at even a pre-cancer stage. I hinted in my talk, we now know a lot of these come from the fallopian tube, and we really need to be directing our efforts at understanding fallopian tube biology and what are the very first steps in the cancer development, because that will give us the key of how we get in with a screening test. But no, it's a real shame that we don't have a screening test at the moment, but at least we did the trial and know that for certain. I think that was really important.
Thank you, Richard. And I think just to add from that, obviously from Target Ovarian Cancer's point of view, this is an area of huge priority for us, understanding more about early diagnosis and looking at what are those opportunities, exactly that, to identify cancer, ovarian cancer earlier. And it's not the end of the road. And I think it's something that we will continue to be focusing on. And when we think about treatments, and clearly you've talked through some of the work that you and your team have been doing, looking at this, what would you say is on the horizon in terms of more targeted treatments for ovarian cancer? What could we be looking forward to? What are the key areas for research at the moment?
Okay, well, the really big development in the last 10 years, and we can't have a talk without looking back a little bit, has been the development of PARP inhibitors, and those are our own targeted therapies. I mean, they are working par excellence in ovarian cancer, so let's be really proud of that a little bit. So that's a great bit of story, but I think most people know PARP inhibitors aren't for everybody, and they don't work in every tumour type, so we do need more targeted therapies. Bevacizumab or Avastin is there as well. That's had less effect, and I think it has a role, but its role is a little bit limited. There's a lot of focus at the moment on the idea of immunotherapy. So what we understand now is cancer is a true battle at the cellular level between the cancer cells growing from the one end and the immune system fighting back from the other end. And if we can supercharge or turbo boost the immune system, can we make a difference to that? There are lots of immunotherapy, is not just one treatment. There are lots of ways of trying to attack immunotherapy. The first line drugs that we developed, called the checkpoint inhibitors, are working really well in some cancers, but then don't seem to be quite as effective in ovarian cancer as perhaps we'd hoped. Going back to my librarian analogy, it does seem that tumours that have one of those defects, the mismatch repair defect, one of those librarians missing, they're probably the best for immunotherapy, but that's quite a small group of cancers overall. So there might be a role for immunotherapy, and there are ways now we're talking about using things like TIL therapy, where we take the white cells, the immune cells from the tumour itself, take them out of the tumour, expand them in the lab, and then put them back into the patient another time. So there are trials coming. And then I think the really neat thing will be, how do we combine some of these things? Can we put PARP inhibitors with immunotherapy or with bevacizumab? And how do we all mix them up? So yes, targeted therapy is already here, but there's quite a lot of work to do to refine it. So yeah, that's on the horizon.
Okay, that's fantastic, thank you. And in terms of thinking of other kind of big opportunities in the future, thinking about targeted treatments, for example, would you foresee a time when having having different testing could help people understand what treatment is going to work best for them?
Yeah, I absolutely. I think where we want to be is a position where, at every time a patient needs treatment, their tumour is being analysed, and the results of that analysis are determining which treatments they're most likely to get benefit from. So I would love to be sitting here in 10 or 15 years time saying that our management of ovarian cancer is really, really complicated. And it's not just that we give carboplatin to everybody, which is sort of what we've done for years, but instead we're able to give drug A to patient A and drug B to patient B, and so on and so on, all dependent on what their tumour is actually saying at that particular time. So I think it's highly likely we'll need to biopsy tumours every time we need to treat them to understand what treatment is going to be the best for them.
Thank you, and absolutely agree with that. I think we all know that research has been impacted by the pandemic, as many of our lives have, and it would be useful to understand, or it'd be interesting to understand from your perspective, what can we be doing now to make sure that we really don't lose the momentum on ovarian cancer research? We've got these potential ideas and breakthroughs on the horizon. What do we need to put in place as funders, as people affected by ovarian cancer? What could we be doing to really make sure that we're accelerating progress now?
I think we can answer that on two levels. So one is on a national level. I think in the last year, everybody has been impressed by the scientific knowledge and expertise there is in the UK. And I think what we need to do is harness that enthusiasm nationally and try and make sure that everybody is very proud of our science background in this country and continues to invest in it. So anything we can do that encourages investment in science in general will be a good thing. And I think, you know, that global idea is very important. On a slightly more niche level related to ovarian cancer, then yeah, we are in for a hard time, because everywhere is trying to get back on its feet. Labs are still recovering a little bit from what we're doing. Clinically, we're all working the backlogs that have inevitably developed over the last year or so. So there's some real challenges, but I think anything we can do now to raise the profile of ovarian cancer, continue to raise money for research, and continue the dialogue. The more we talk about it, the more we get the community behind us, the better we'll recover from this and get things going again. So yeah, events like this are going to be really important to try and just up that agenda.
Great, thank you. Unfortunately, it's I'm going to have to draw it to a close because we're coming up to 2 o'clock, and I don't want to take up people's time this afternoon, but just wanted to say a huge thank you, Richard, for taking on those questions, and apologies again if we weren't able to pose your specific question. Please do give us a shout, we will be able to come back to you on those.
Absolutely no problem. There's some really good questions come up in the chat. Will we have a chance to feed those back, do you think, at some point?
Yeah, we absolutely can. If you're happy to take those away, then what we'll do is we can record those and make sure that we get back to you. I'd also just like to say again to anybody who, if anything that's come up today that has given you cause for concern, specific questions you wanted to talk through, please do give our support line a call. So it just leaves me to say a massive thank you to our speakers, to everyone who's made the event today happen, and also for all of you for joining us this afternoon. We've had an absolutely fantastic turnout, you know, lots of people joining us. I know this is an area of huge interest for everybody. Our research programme is only possible thanks to the incredible generosity of all of our supporters and partners. I know many of you are on the call today, and to the dedication and commitment of our research advocates, so a huge shout out to you as well, and the ovarian cancer research community, the teams up and down the country who are really taking us to the next stage in ovarian cancer research. So please take our thanks for everyone's support in the success of our programme today. Clearly we're not resting on our laurels. We know there is plenty more to do, and we were really delighted, thanks to your generosity, to launch a new round of grant funding just a few months ago, so that we can continue to strive for new and better treatments for ovarian cancer. We will be sending out more information after the event. We'll do our best to answer questions where we can. If you've been inspired through this to join our Research Advocate Network, we'll also send a link around for this. So do find out more about it if that's something of interest. And we'll be sharing a short survey as well. We'd be really grateful if you could feed that back to us. We want to run more of these kinds of events in the future, so it's really helpful for us to understand what's useful, what's interesting, what's relevant to you. So please don't hold back in your feedback. Remember, if you'd like more opportunities to speak to others who are affected by ovarian cancer and experience more of our digital events, if you've not done so already, please join the ovarian cancer community. This is an open and understanding Facebook community for everyone affected by ovarian cancer anywhere in the UK. And as a final plug, again, if you've got any queries or questions, our support line is always available by phone and email, and those details are in the chat. So I'm going to finish there and say thank you again so much for joining us today. It's been absolutely fascinating, and please enjoy the rest of your day. Thank you very much.